Okay, good morning. Right. So in the previous we have discussed about growth and its different factors and all the parameters we are measuring the growth, right? So let us coming to another one important thing with respect to the plant is development. What do you mean by development? So development is nothing but simply we can define in only two terms. It is sum of very good. So differentiation is nothing but development is nothing but growth and differentiation. The sum of growth and differentiation can be termed as development. Okay. So development is nothing but overall the total changes that have been taken place from its birth to the death. Andre on the life period alli enella changes agutte, all the changes taken place throughout its life. So ಆ ಒಂದು changes ಅನ್ನು ಅಂತ ಕರಿತೀವಿ development. So what do you mean by development? So development is nothing but all the changes that takes place throughout its lifetime. That means it is nothing but it is a sum of both growth and differentiation. We know that growth is nothing but increase in number of cell or in a mass, that is by the division, but it will get its definite structure and function by it attaining the maturity, that is what differentiation. During that we will see it is the sum that what we call development. So development is all the changes that occurs during its lifetime, that is all throughout its lifetime, right? Okay, so how we can see that? So development andre agar growth and differentiation andre, see here if it is a seed will get germinated, the seed will get germinated means it will give such the seed germination, that will give such the seedling. This seedling will get enhances its growth, that is what elongation, and then maturation, so that when it attains the maturity that will give such to what reproductive structure. That reproductive structure in the plant is called flower, so that will lead to the flowering. When the flower is get developed and completely involved in the fertilization, that will lead to the fruit, but ali directly seed ಕೊಟ್ಟಿದ್ದಾರೆ book ಅಲ್ಲಿ, okay ತೊಂದರೆ ಇಲ್ಲ. So flowering too, it will give rise to seed through the fruit. So this seed now enters into the aging process, that is called senescence, and then it leads to the death of an individual plant. So from beginning with the seed, then seed will get germinated under some conditions, because there may be dormancy or maybe there is a pause or quiescence irbodu ಅಥವಾ dormancy irbodu, whatever that, whether intrinsic or extrinsic, ಎರಡು ರೀತಿಯಲ್ಲೂ ಕೂಡ it will be get stopped. So seed either it is gets ultimately the germinated, it will give you the seedling that will get elongates and develops the normal vegetative phase, that is what juvenile phase. That is juvenile phase is nothing but what vegetative phase, where it will get the body weights or body growth is completely seen, then it will lead to the maturation. Once it accompanied its complete vegetative growth, then it enters into the maturation. So if it is without completion of the juvenile, if it attains the maturation, it is early development, it is incomplete, it may be the immature embryo it developed, or it may be not leads to the fertilization, because if the flower structures are not completely developing means there is a reason there are vegetative structures are not completely developed. Hagagi there is a requirement of complete involvement of the juvenile phase, the fully completion of the juvenile growth is seen means vegetative growth is completed, then it attains the reproductive maturity. Once the reproductive maturity is attained, it will bear the reproductive structure. The reproductive structures will involve in the reproduction, that will give such the product that ultimately gives to the next generation by this one. But after giving the seed, not the seed undergoes senescence here. Aroma kilcotina, it is not the one. Once the seed is beared on the plant, that plant, especially if it is an annual plant, that will enter into senescence phase and leads to the death. If it is perennial, it will give us the repeated flowering and repeated fruit setting and seed setting, and after some years it will get die. That is what the thing which varies from biennial and perennial plants. So likewise, from a seed to a death, that means from one initiator to the one complete life end agode yav riti andre birth to death annada indicate ಮಾಡ್ತಾ ಇದೀವಿ. Clear? So likewise, whatever the changes as we are seeing here, from a seed to till its death, means from germinated seedling to the death by attaining the senescence, natural conditions anna ಮಾತಾಡ್ತಾ ಇರೋದ್ರಿಂದ this all changes observed throughout its lifetime is development. Okay, as we know the phases of growth, there is meristematic, maturation, and in between we can see elongation phase. The same way here, the development is seen through the meristematic cells. So this meristematic cell, okay, which enhances its what? Protoplasmic content. Okay, the protoplasmic content is increased, nothing but what? It is plasmidic growth, right? So the plasmidic growth means increase in the protoplasmic content is called plasmidic growth. Then here it is nothing but it led to what? Elongation. Okay, then that involves in maturation. Nothing but it is giving rise to the attaining of specific function and specific structure. Specific structure, definite structure is assigned to that particular cell which have been elongated, and ultimately it gets matured and become permanent. That is what? Differentiation. Now these permanent cells, see, from meristematic cells to what? Permanent. We have seen that it will enters into death by attaining senescence. So what is this one? This is death. See, from birth, that is the actively dividing cells which increase their protoplasmic content, that is called the plasmidic growth. So once the protoplasmic content is increased, the cell will get elongated, that means it will bulge out. That is what? Elongation. After attaining the weighted growth, they will attain the maturation, that is what? Reproductive maturity. So after the maturation, we will see, that means they will be getting definite structure and it's assigned with the definite function, that is what? The differentiation. That is what we call the plant will undergo self perpetuation. Once the cells are loses their division property, they will become what? The permanent one, right? So these permanent cells will be carrying out the function of the plant up to when they get the sufficient amount of food and water nutrient availability, but after some period of time they will get aged, that is what? Senescence. So due to the senescence, the metabolic rate will get decreases and ultimately it will ends up that ending of that metabolic rate or metabolic activities in the plant leads to death of an individual. Likewise, these changes will be notified. But here think, the plasmidic growth, whichever the cell which increases cytoplasm, this is one kind of thing, right? When the cell is increased its cytoplasmic content, it will lead to what? Either differentiation or it may undergo again the division, because to balance the nucleocytoplasmic ratio, the volume which is present inside the cell, that is in the cytoplasmic content and also the nuclear content, they need to be in a same ratio, means in a balanced ratio. So in average cell ಅಲ್ಲಿ ಯಾವ ರೇಷಿಯೋದಲ್ಲಿ ಇರುತ್ತೆ ಆ ರೇಷಿಯೋಲಿ. That's what in the plasmatic growth, that's why the cells will undergo further de-differentiation to develop the new cells by undergoing meristematic property. Likewise, a whole individual throughout its life will undergo these changes from growth and differentiation. That changes are nothing but what? This one. Clear? So likewise, we can see the developmental activities in a plant life. Clear? But there are some plants. Is it clear? Okay. So in some conditions, the plant will follow different pathways. That is, the plant follows different pathways with response to there is a main term environmental conditions and also with response to the its phases of growth. This response means with respect to of environmental condition or with response to of its phases of the growth, the plant is following the different pathways. That is nothing but plasticity. What do you mean by plasticity? Means if a plant follows a different pathways in response to environmental conditions or phases of growth. What are the environmental conditions? Whether it is terrestrial, whether it is aquatic, whether it is aquatic submerged or freely floating, okay, whether it is underground, whether it is aerial or subaerial, these are all what? The different conditions of the environment which the plant is living. The same means based on those conditions, the plant will show different structures. That means here by following these different pathways, it forms what? Different structures. The plant will follow the different pathways with response to the phases of growth and environmental condition and forms the different structures in the plant body. So that different structures are formed what? Best example we can take here is heterophily. What is hetero? That is different. Phylla refers to leaf, bearing of two different leaves in the plant body, whether in different environmental condition, whether it may be in the different phases. Means in an early condition it is bearing one type of leaf, in a mature condition it is bearing another kind of leaf. That is the developmental conditions. That means the phases of the growth. That heterophily is called developmental heterophily. Okay. So here heterophily is taken as a best example. With respect to environmental condition, we are having the plant called ranunculus. What is that? Buttercup. Common name is buttercup. So as per NCERT, the given plant name is buttercup plant, which shows the two different types of leaves. When it is in a terrestrial condition, it bears one kind of leaf. When it is in an aquatic condition, it bears another kind of leaf by making the different incisions. See, when a plant is in a terrestrial condition, it bears this kind of leaf. When it is in a terrestrial. When the plant is in terrestrial condition. This is what? With response to environment. Because terrestrial condition I am saying, right? So, that is in a aerial one. Now, it is in aquatic condition. It is the same plant. That is the ranunculus. That is what the buttercup, which is present in an aquatic condition. We can see these leaves, which we have seen. What we have seen here, as the lamina, that is the outer margin or a leaf blade, what you are saying, it is not so incised, that is incisions, there is a inward projections in the lamina. So, idu lamina higide andre, there is no inward projections like this. But here the incisions are more to adjust with the water condition. Allirtakanta water current ge adjust agbeku. That is what here the thing. When they are in terrestrial condition, to avoid the wind velocity, they are bearing such leaves, especially in the terrestrial condition, they are bearing less incised leaf. When they are in aquatic condition, the same plant which is there in an aquatic condition will bears the different leaves from the terrestrial by getting more incisions. Ivala enivu incisions. This is what? Difference in the leaf observed in the same plant present in the different environment. One is terrestrial, another one is aquatic. Second thing is developmental plasticity. That is what heterophyly. Developmental heterophyly is what? At different phases of the growth. As we know the three phases, meristematic, elongation and maturation. Here, when the plant is bearing normal leaf in an early condition, but the same plant will be having another type of leaf in the same plant body when it is mature. NCERT ಅಲ್ಲಿ ಇದನ್ನೇ ಸಪರೇಟ್ ಈ ಕಡೆ ಬರೆದು ಕೊಟ್ಟ ನಾನು ಒಂದರಲ್ಲೇ ಬರ್ತೀನಿ, ಒಂದೇ ಪ್ಲಾಂಟಲ್ಲಿ ಇರುತ್ತೆ ಅಂತ ಗೊತ್ತಾಗಲಿ ಅಂತಷ್ಟೇ. ಇಲ್ಲಿ ಡೆವಲಪ್ಮೆಂಟ್ ಅಂದರೆ ಏರಿಯಾ ಚೇಂಜ್ ಆಗಿ ಇಲ್ಲಿ ಎನ್ವಿರಾನ್ಮೆಂಟ್ ಆಗಿರೋದ್ರಿಂದ ದ ಪ್ಲಾಂಟ್ ಈಸ್ ಶಿಫ್ಟೆಡ್ ಟು ಡಿಫ್ರೆಂಟ್ ಎನ್ವಿರಾನ್ಮೆಂಟ್ಸ್. ಒನ್ ಈಸ್ ಟೆರೆಸ್ಟ್ರಿಯಲ್, another one is aquatic. But here the same plant, in the same plant we can see the two type of leaf. The younger leaves are showing no incisions, there are less incisions, but the mature leaves are showing more incisions. What is that? During the plant growth phases, in early phase it is showing the less incision. When it is having the mature, it is showing the more incision. That what? In the same plant we can see there is a changes with respect of the development. That's why it is called as developmental heterophyly. The best seen in coriander, cotton, larkspur. So these are the different example for plasticity with respect of environmental condition and with respect of developmental conditions, developmental phases, means plant growth phases, right? Clear as to? So this is how the plant is giving response to the different conditions by following the different pathways, developing the different structures with respect of environment and its phases. But what are the conditions which makes the plant to get developed like this? That is what we have studied. The conditions for growth and the enala nodadvi. Here also the same, the conditions for development. That is extrinsic factors and intrinsic factors. Let's see that one. Is to clear idiyana? Okay. conditions for growth. Okay, so here conditions for development. Okay, so the plant development is mainly based on two factors: extrinsic factors and intrinsic factors. What are the extrinsic factors? That is, they are dependent on external factors. The external factors are light, temperature, water, oxygen, nutrients. Right? Whereas the intrinsic factors of the plant are developed inside the plant, that is, are of two types. One is inside the cell, other one is in between the cell. So which is present inside the cell? That is the genetic control, the genetic composition which is making the development. ಯಾವ ರೀತಿ ಆಗಬೇಕು, ಅಗರ್ ಇಲ್ಲಿ ಜೀನ್ ಕಂಟ್ರೋಲ್ ಮಾಡ್ತು, ಅರ್ಲಿ ಸ್ಟೇಜಲ್ಲಿ ಈ ಥರ ಇರಬೇಕು, ಲೇಟರ್ ಸ್ಟೇಜಲ್ಲಿ ಈ ಥರ ಇರಬೇಕು ಅಂತ. ದಟ್ ಇಸ್ ವಾಟ್ ದ ಜೀನ್ಸ್ ವಿಚ್ ಆರ್ ರೆಗ್ಯುಲೇಟಿಂಗ್. ಆನ್ ದ ಸೇಮ್ ವೇ ದೇರ್ ಮೇ ಬಿ ದ intracellular intrinsic factors. Okay, the intracellular intrinsic factors are plant growth, regulators. These plant growth regulators are of two types. They are promoters and inhibitors. But in this chapter we are going to discuss about these PGRs more. Clear? So before going to that, we know that how light is affecting. Agare light annodna navu ಈಗ ಮೇನ್ ಆಗಿ ಮಾತಾಡಲೇಬೇಕಾಗುತ್ತೆ, because we know that etiolation. What do you mean by etiolation? In the previous only we discussed etiolation means development of pale creamy colored plant body. There is a non-green structures because the chlorophyll pigments are absent, they are not synthesized, because chlorophyll synthesis requires inducing by the light. Photoinduction is required, right? That is what etiolation. That is enhances what? Plant shoot system. Length of the shoot, that is a plant shoot system is enhanced. Length and increase ಮಾಡುತ್ತೆ. That is what, with response to the light we can see etiolation. Means etiolation is found what? Not in presence of light, means if light is absent. As I mentioned in the previous, if you can see the germinated seeds, they have not came in exposed of the light, they will be showing etiolation. Means once the seed which have been under the darkness, they have been germinated, then they will be showing etiolation with no chlorophyll presence, with more or enhanced shoot length. Okay, that is what we can see here. That is etiolation. There is non-green part with enhanced shoot length will be shown only in what? Darkness. Clear? But in presence of light, it will produce chlorophyll, it will act as a green, and automatically the coleoptile will get differentiated, and ultimately that will give such the leaf and that will be functioning as a photosynthetic organ. That will be independent. Likewise, the temperature, water, oxygen, nutrient, that is availability of nutrient is important, and oxygen, again ನೀವು girdling experiment ನೋಡಿದಿರಿ, right? So nutrient it has been stopped to reach the sink, then automatically the sink will die first, and then later the source will also get died, especially with respect to sink as root. Then oxygen availability, that is complete oxidation of complex organic compound that leads to the release of more energy. And water, metabolic content and metabolic activities are mainly dependent on water presence. Cytoplasmic volume or a protoplasmic growth, nothing but plasmatic growth, that is all dependent on water. Enzyme activities will be shown, and for that optimum temperature 25 degree to 30 degrees Celsius, that is also the temperature which is required for a normal growth of the plant body. Clear? So these are all the different conditions and that the plant development will be dependent on. This is all about the extrinsic, but whereas in the intrinsic one, as we have said, one is a genic control, that is the genetic predisposition which is already there in the plant body. Predisposition already means before there is a deposition of some genes. Based on the genes, even though we will be giving these all conditions, that will not get the proper development because the gene is present. For example, we can see if there is a homozygous recessive for a pea plant, the tallness is a factor, the allele of T which is homozygous recessive. Homozygous recessive means what? Right? Then we can't make that normal without any applications. Then how can we make? By plant growth regulator we can make, or we can overcome that one. That is what genetical dwarfness can be overcome by the intercellular intrinsic factors called plant growth regulators, especially gibberellic acid. That is one example we can quote, right? So that enhances the genetic dwarfness. That is what a bioacid. Okay, so let's see with respect to of plant growth regulators. Ili tanka clear idina? Okay. ಇಷ್ಟು ಅರ್ಥ ಆಗಿದೆಯಾ? ಓಕೆ. Coming to second phase of this chapter, that is plant growth regulators. So as the term which is regulator we are mentioned here, it is either enhancing the growth or maybe pausing the growth, or it may be inhibiting or completely stopping the growth. Enhancing and arresting and completely stopping, that means triple effect. Muru ide. So they are all the plant growth related regulation. That is why those chemicals which regulate these plant growth, they are called plant growth regulators. As we know, they are also called as plant growth hormones. They are nothing but phytohormones. These are the chemicals, they are organic chemicals, especially non-nutrient organic chemicals which are produced in the trace amount but alters the plant growth activities. Okay, they are non-nutrient, ಅದಕ್ಕೆ ನೇರವಾಗಿ ನಮಗೆ ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಆಗಬೇಕಾಗಿಲ್ಲ ಅವು. They are non-nutrients ಆಗಿರಬಹುದು, and they are present inside the site where they are produced will be leads to the site where actually the activity is required. Means site of synthesis is different from the site of activity. That is where the action is shown, means its activity is exhibited, right? So like that, the phytohormones are such chemicals which are required in a very less amount or a tracer amount, as you have studied hormones in chemical coordination and integration, right? The same here also the hormones, but only exocrine and endocrine you have seen, right? So here These are phytohormones. But as we mentioned already here, one which is promoting, another one is inhibiting. But some hormones are showing the both effect. Some hormones are showing a promoting, inducing and inhibiting and also arresting. That is triple effect anta kuda some way we will refer. So that is what the phytohormones, which are plant growth promoters, one which enhances the activity like cell division. Cell division is a plant growth promoting activity. Cell dividing one, that is plant growth promoting activity. It is bearing the root, bearing the shoot, developing the structures on the root and developing the structures on the shoot. That may be root hair formation. That is also one activity. Shoot development, that is what? Formation of leaf, formation of fruit, formation of flower, formation of seed, then seed getting germinated. So these are all the activities which are making the promotion of the growth. But what is that one which is making the inhibition? That is inducing or bringing the dormancy. So one which hormone or a chemical which brings the dormancy or which is making what? Arresting. Even though there is a presence of favorable condition, the seed is not getting germinated because of its genomic control. So there is a requirement of particular things. That's what here the dormancy is showing inhibition or senescence. Bringing the senescence is also stops the growth, right? So which brings the senescence or which bringing the dormancy to the seed or a bud, they are called inhibitors. Okay, but there are some which will show the triple effect. That is initiator, nothing but along with that it is promoting. Okay, it is pauses, that is what this one, and also it is arrests, nothing but stops or inhibits. So let's see what is those hormones, which are those hormones making these all different effects by giving rise to the cell to get divide or forming the root or other accessory structures or shoot or its accessory structures called leaf bearing, fruit bearing, flower bearing, seed developing or the seed getting germinating. These are all the growth promoting or growth enhancing mechanisms mainly controlled by the hormones called axin, gibberellin and cytokinin. Okay, so these are the three hormones which acts as promoters. But one which is acting as only inhibitor hormone, which is bringing the senescence, bringing the dormancy, and it is also called as stress hormone, abscisic acid. Especially we can also by this we can say closing of stomata, right? Closing of stomata, that is what? It is acting as a stress hormone. Under unfavorable condition, this is making the closing of stomata. Okay. Triple effect. It is promoting or initiating, means inducing. First one initiate ಮಾಡುತ್ತೆ. Already initiate ಆಗಿರೋದನ್ನ ಮತ್ತೆ it is promoting, or sometime it is making pausing or stopping, and again it can make the resuming or completely stopping. That is what making the senescence. That is what inhibiting. That is the main hormone called the gaseous hormone is ethylene. That shows the triple effect or double effect. Both acts as promoter, and in NCERT, as per NCERT, it is showing the dual nature of hormone, both promoter and inhibitor. Because hormones are mainly classified as what? Promoter and inhibitor. Hence, it shows the dual nature. It brings the triple effect. That is none other than the gaseous hormone called what? Ethylene, right? Okay. Let's see about these hormones and their discovery with respect to of their precursor molecules, and then about their nature. Because these hormones, as I said, they are intracellular, they are produced at one site and they shows their action at the another site. Because of that, they are derived from different components. So, once we can see the indole ring, where we can see the indole ring, that is what we can see the phytohormone called auxin. The precursor here is amino acid, tryptophan. The tryptophan is an amino acid which is acting as a precursor molecule for the development of auxin. Okay, which is containing what? Indole ring. Hence, auxin is a derivative of indole compound. The best example we can study is indole acetic acid, indole 3 acetic acid, or indole 3 butyric acid, IAA, IBA. These are the natural forms. This is one thing. This is hormone. These are the derivative. This is the precursor. Means both are same. Then second thing, we can get this one by terpenes. As per this NCERT mentioned, they are terpene derivatives. Especially they are derived from diterpenoids. The precursor here is acetyl coenzyme A, that is producing an hormone called gibberellin. Example is GA2, GA1, but most used and studied, intensively studied is GA3. Okay. Next, third one, that is derivative of purine. It is nothing but modified purine, mainly by the adenine. Another purine is guanine. Adenine and guanine are the purines, but here it is a purine derivative that is developed by N6 porphyryl amino purine. That is nothing but kinetin. Later it came to know that it is having the property of cell division, hence it is called cytokinin. Hence we can get the cytokinin by sperm DNA, that is herring sperm DNA, or even in the tRNA derivatives, that is also that one, because DNA or tRNA both are having the purine, both are having adenine. Hence from the adenine modification we can get the N-porphyryl aminopurine, nothing but the kinetin, that shows the cell dividing property, that is called cytokinin. Like that we can get this one. Clear? Then fourth one, there is no such thing which is only showing the gaseous property. C2H4, that is methionine compounds. From the methionine compounds, that is also amino acid, right? AUG, right? So from that we can derive this called hormone is ethylene. Commercial form it is available is ethaphone. Okay, etaphone or ethaphone, what we refer. Okay, then last one, abscisic acid. How we can get the abscisic acid? Abscisic acid ಯಾವುದರಿಂದ ತಗೊಳ್ತೀರಿ? Huh? Other? It's, it is a derivative of the auxiliary pigment in the plant called carotenoids. It is a carotenoid derivative ಅಂದರೆ xanthophylls ಅಂತ ಏನೆಲ್ಲ ಮಾತಾಡ್ತೀವಲ್ಲ, ಸೊ ಆದ್ದರಿಂದ carotenoids ಅಂತ ಏನು ಕರಿತೀವಿ, ಆ carotenoid derivative that is abscisic acid obtained in the form of this one. So these are all the different phytohormones, but all these hormones which have been discovered are not intentional one, they are accidental discoveries. ಏನೋ ಒಂದು ಡಿಸ್ಕವರ್ ಮಾಡ್ಬೇಕಾದ್ರೆ ಇನ್ನೇನೋ ಒಂದು ಸಿಕ್ಕಿರೋವಂಥದ್ದು. ಡಾರ್ವಿನ್ ಫೋಟೋಟ್ರೋಪಿಸಮನ್ನು ಸ್ಟಡಿ ಮಾಡಬೇಕಾದ್ರೆ ಆ ಕೆನರಿ ಗ್ರಾಸಿನ ಕೋಲಿಯಾಪ್ಟೈದಲ್ಲಿ ಇದನ್ನು ಕಂಡುಹಿಡಿತಾನೆ. ಆನಂತರ FW ಇವೆಂಟ್ ಬಂದು ಅದನ್ನು ಐಡೆಂಟಿಫೈ ಮಾಡ್ತಾನೆ. ಗಿಬರ್ಲಿನ್ ಯಾವುದೋ ಒಂದು ಪ್ಲಾಂಟಿನ ಸೀಡ್ಲಿಂಗ್ಸ್, ದಟ್ ಇಸ್ ವಾಟ್ ದ ಪ್ಯಾಡಿ ಸೀಡ್ಲಿಂಗ್ಸಲ್ಲಿ ಇರ್ತಕ್ಕಂಥ ಬಕಾನೆ ಡಿಸೀಸ್ ಆರ್ ಫೂಲಿಶ್ ಡಿಸೀಸಲ್ಲಿ ಆ ಫಂಗಲ್ ಡಿಸೀಸನ್ನು ಅಥವಾ ಡಿಸೀಸನ್ನು ಸ್ಟಡಿ ಮಾಡಬೇಕಾದ್ರೆ ದೇ ಕೇಮ್ ಟು ನೋ ದಟ್ ಇಟ್ ಕೆನ್ ಇನ್ಕ್ರೀಸ್ ದ ಲೆಂತ್, ಇಟ್ ಕೆನ್ ಇನ್ಕ್ರೀಸ್ ದ ಶುಗರ್ ರಿಲೀಸ್. Then they came to know about the gibberellic acid extraction. Now we are having hundreds and more than that of gibberellic acids. Gibberellins namaliga we are having more than 100 types. Then purine derivative, while studying the tobacco plant diseases, they came to know that actively dividing cells, agar alli actively dividing cells which are that they contain a kinetin, that is what this one. Later the same thing was identified in the corn's kernel, maize corn kernels ali, and even in the coconut water we came to know about this one, that is what cytokinin, that is actually we use in the Mirashige-Skoog media or Miller-Skoog media, MS-Skoog media, what we use in the tissue culture called MS-Skoog media, that is a specialized media. It is having a BAP, that is what benzyl aminopurine, nothing but zeatin, commonly called as zeatin. Zeatin is nothing but benzyl aminopurine. Okay, likewise we got synpurine. The same way the gaseous hormone, which one, the fully ripened fruit which is containing that one, the banana especially, but the first discovery was based on the orange fruit, where the fully ripened, that is what we call rottening. The fully ripened fruit is nothing but, completely ripened is nothing but it is at the end of, means it is at the stage of rottening, ಕೊಳ್ತೋಗೋ ಥರ ಬಂದಿದೆ ಅಂತ ಅರ್ಥ. ಅಗರ್ if you want to eat the ripened fruit, you must eat which one? You need a ripened fruit means what it contains? Highly ethylene. High ethylene is found at what? It is in a end of ripening, nothing but at the initiation of rottening. That is what it contains, ethylene. That is what gaseous, that is volatile compound. They came to know about that. That volatile compound was induced the ripening in the other unripened fruit near to them. Then they came to know about that volatile compound. That was the discovery of the gaseous hormone called ethylene. Okay, there are some conditions in the plant where we have seen the carotenoid rich contents, but they are not leading the seed to get germinated. Then they came to know that there is a compound called the abscisic acid which is making the dormancy and which is bringing the closing of stomata. That is what it is present in a stress conditions. Like that, the discovery of these all the phytohormones is not actually the intentional, it is just a accidental discovery. Clear? So in the next session we will discuss about all these promoters, triple effect or a dual nature, and inhibitor hormone with respect to of their discovery, their derivatives as we already known, and their action, site of synthesis, and their physiological and applications in a horticulture or plant growing. Clear? Right. Thank you. Take care.